Good afternoon. This is obviously Authentic Biochemistry Podcast video. Today's 26 May 2023. And I've been waiting a long time to do a video, and I decided to break down and do it today. Let's just get started. Now, what I'm going to discuss with you today is one of these old-fashioned physiological biochemistry lectures, because it follows in suit from what I was talking about just yesterday, again discussing lipoprotein metabolism, the regulation of components that involve both pathophysiological and normal physiological states, such that lipoprotein metabolism is tightly regulated at multiple levels: the level of expression of the genes, that is transcription, translation of the polypeptide, mobilization of the various proteins subcellularly to accommodate the multiple organization around synthesizing lipoproteins, which, remember, are non-covalently associated lipid and protein. Proteolipids are covalent association of lipid to protein. And we've talked a lot about those: acylated, prenylated, etc. proteins. In fact, recently we talked about octanylated protein, ghrelin. That would be basically a proteolipid, because it is a protein that has a covalent modification of a lipid. Lipoproteins are not covalently bound, but the assembly of lipoproteins is very complex because it involves a certain kind of mixing that requires always an emulsion state, so that the acyllipids can move to the center of the lipoprotein complex because they are shunning water, the hydrophobic effect, and increasingly from the core of the lipoprotein out to the surface, you're going to have increasing amounts of hydrophilicity. And we already talked about the architecture of that and the density and the size and whatnot. So we're going to get to that, but I want to give you the basic foundations of digestion. That's what we're going to talk about right now, a little bit of hormonal regulation. And then, of course, finish that suite of lectures today, and maybe I'll do another one tomorrow, maybe both video, and get a couple of hours like that under our belts. And then we can go back into the audio version where I'll get back to the primary literature. Okay. So I want to just get, finally, that was, I think, way longer of an introduction than I thought I was going to do, but I'm always like that. I always do a lot of, right off the top of my head, try to figure out what I want to say to introduce this lecture. And that's what you just experienced. All right, so let's go. All right, so this is a slide I've used several times in lecture. It's an excellent one. It comes from one of my favorite textbooks. Now, remember, when I taught biochemistry at university in a lecture hall, I didn't use a textbook except as a reference. And some of the textbooks are really excellent, and some of them are not so good. This particular one I used in medical biochemistry and veterinary science biochemistry, and also in some physiological components of pharmaceutical biochemistry lecture courses. Okay. And of course, nutrition, exercise physiology, etc., and general biochemistry graduate school. Okay. But the Devlin text I'm getting these slides from is better for biomedical application. Other biochemistry texts, such as the Vote and Vote text, is much better for dealing with the nitty-gritty of enzymatic reactions, particularly a very well written series of chapters in that text on kinetics, thermodynamics, and regulation at the level of protein interactions and modifications of proteins with lipids and carbohydrates. The real earthy sort of biochemistry that I do a lot of discussion with when I do the audio. So You see, when you digest food, you're going to have the three major food groups. You're going to have carbohydrate, you're going to have protein, which will be broken down to amino acids in the gut because of proteases, endoproteases, and exoproteases, specific for the proteins you're digesting, chymotrypsin, trypsin, things like that, and then a whole host of others. The lipid is going to be broken down. Remember, we talked about lipases. So you have triacylglycerol lipases reforming TAG into DAG and MAG. And the process of removing fatty acids from the glycerol backbone, which is the major source of lipid in the diet. Of course, you're going to have some phospholipids too. But the idea, the system works in the small intestine to take the dietary triacylglycerol, hydrolyze it, and then resynthesize using components of the Kennedy pathway. So the triacylglycerol is the correct molecular event, that is physically and chemically, to make the emulsion to form the chylomicron, which is occurring in the small intestine, right? So that's why there's a retailoring of the tag, and you don't just get the tag right directly from the diet. There's a lot to discuss there. I'm just giving you that very brief understanding of it. So because you're going to have carbohydrate in diet typically, postprandially, you're going to have the beta cells of the pancreas secreting insulin because glucose levels are going to be surveilled by the beta cells. And lipid also is going to play a role in the secretion of insulin. We've talked about this in the past, and I will detail it soon. Once insulin is secreted, then it's going to go to the portal vein along with the digestion of carbohydrate, protein, and lipid. Now, the lipid will go, the chylomicron that's formed will go directly into the lymphatics. And you can see that once that occurs, the chylomicron can deliver its cargo of lipid to skeletal muscle, to adipose, and also directly and indirectly to the liver, right? That's important to understand because as soon as the diet introduces triacylglycerol, and that's the major component in terms of caloric density and overall mass of the amount of lipid you're going to be consuming, right? You're not going to be consuming great amounts of cholesterol or cholesterol ester, even when you're eating animal fat. And when you do, you also have a lot of animal muscle, much more muscle than fat typically. And that means you're going to have a lot more protein than you are actually having the membrane components of mammalian membrane, of mammalian cholesterol content of those membranes. Okay, so you're going to be delivering triacylglycerol to synthesize intramyocellular triacylglycerol oil droplets in skeletal muscle and also in cardiac muscle. You're going to be delivering a lot of that triacylglycerol directly from lipoprotein lipase mediated transfer to the adipose, growing that adipose from the chylomicron. You're going to generate during these processes chylomicron remnants because they're going to be smaller, right? And they're going to be increasing in what? In density, because there's going to be less lipid, right? More protein, less lipid means you're going to have more density. And those are going to circulate back and bind to their receptors in the liver. And likewise, unloading some of the cargo lipid from skeletal muscle will also make it to the liver via that chylomicron remnant pathway. Okay. Amino acids, you can see when they enter the liver, can be used to resynthesize polypeptide. The glucose can then be converted to, via glycolysis, triacylglycerol via pyruvate dehydrogenase, pyruvate carboxylase, the TCA cycle, to citrate, ATP citrate lyase, generating acetyl-CoA into the cytoplasm, then for the synthesis and storage of hepatic triacylglycerol, phospholipid, and of course cholesterol and cholesterol esters, which are going to be leaving the liver subsequent in the form of the very low density lipoprotein. Some of the carbohydrate glucose will be converted to the polyglycan alpha-1, 6, alpha-1, 4 linked homopolymer glucose. Some amino acid degradation will occur, so some urea will be generated in the liver. Now, what's going on in terms of carbon entering the various organ systems of the body, you know that you're going to have primarily glucose is the most significant of the carbon source to synthesize ATP. So most of the bioenergetics in the brain, because of the blood-brain barrier, you're not going to get a lot of lipid transferred to neurons, for example, from circulation. You will get some apolipoproteins, apolipoprotein E we've talked about before, which has a conduit association, very indirect, to certain components of prodromal Alzheimer's disease. ApoE, that's in the HDL fraction, by the way, not the LDL. But the brain primarily is doing all of its work by glucose uptake, because glucose is very water soluble, so it's easy to make it into the neurons, and of course the glial cells in the central nervous system. Now that changes as you move away from the digestion process. You start getting into the postprandial utilization of carbohydrates such as glycogen, and then some amino acid metabolism can occur, and ultimately you're going to be using storage depot fat, and that's going to require the reduction of triacylglycerol from the adipose tissue via mobility from the adipose to the liver, and then lipoprotein lipase is delivering fatty acids to the liver. The liver is esterified to coenzyme A in the cytoplasm of the hepatocyte, and then moving that acyl-CoA via synthesizing acylcarnitine, first in the outer, then the inner mitochondrial membrane, translocating back into now the acyl group will be then re-esterified to coenzyme A within the matrix of the mitochondria, and that acyl-CoA then will go through beta oxidation. Beta oxidation will produce acetyl-CoA. Acetyl-CoA can be used to synthesize the two ketone bodies, acetoacetate and beta-hydroxybutyrate. Right? So I just want to put all that in there now because it's not on the slide. You can see that VLDL will be secreted from the liver, and it's going to be trafficking the same triacylglycerol phospholipids, both glycerophospholipids and sphingolipids to some component. Apolipoprotein load will also be there. There'll be a lot of transfer of those apolipoproteins in circulation, as we've been talking about. This is in the well-fed state when insulin is causing lipid uptake in the adipose and the muscle, as well as causing that is related to spontaneously inducing the expression of receptors on the surface of adipocytes, on myocytes, two examples, right? Such that lipid can be transferred. Right? We talked about low density lipoprotein. We talked about fatty acid receptors like CD36. You get what I'm talking about. Okay. Onward. Early fasting, what's happening? Nothing's coming from the gut because you've done all your digestion. Pancreas starts to recognize low levels of circulating glucose, so no longer insulin being secreted, but glucagon. That enters the portal vein. It's going to start playing a role. First of all, you're going to get glycogen breakdown to glucose. You can get some glycolysis to lactate, right? But the skeletal muscle is going to be functioning before lactic acid is actually synthesized via lactate dehydrogenase from pyruvate after glycolysis. So the liver will not be actually generating a lot of lactose. There won't be a lot of lactogenesis in the liver during the early stages of fasting. But you are going to get lactic acid produced because of glycolysis, ending with pyruvate, then to lactate via the lactate dehydrogenase. And that lactate, because of the Cori cycle, remember, it's going to be sending lactate up into the liver. Then you're going to get gluconeogenesis. So some of the carbohydrates going to be necessary for making the bioenergetics function in the central nervous system. It's going to be glycogen breakdown to glucose. Glucose then sent back into circulation to the brain, and you're going to have complete combustion of carbon dioxide and water there. You're not going to be involving too much of the adipose yet, but you're going to be developing this gluconeogenic substrates, both lactate and alanine, because alanine, once transaminated, will make pyruvate directly into the liver cells. Okay, we won't worry about what's going on in red blood cells right now. It's basically the same kind of combustion that's going on in the central nervous system, although there's a very active oxidative positive phosphate shunt as well, not just glycolysis. And the reason for that is because you need those reducing equivalents to remove molecular oxygen-derived reactive oxygen species, which build up in red blood cells. So in the major last, say, one day, 2 days after a meal, when you're going from fasting to long term leading into starvation state. So remember that human evolution involved a hunter gatherer diet, which meant, and this would have been also when you think back about the early evolution of the biochemical pathways in humans, in that particular mammalian species, which is us, before the understanding, I won't say invention, but the discovery of how to make fire. And of course, before there was any such thing as refrigeration, although because of the seasonal effects, depending on where you lived on the planet, some food could be kept frozen. By and large, the human being went out, hunted, um, digested the food that they hunted, and then carried that digesting process all the way through long fasting periods, right? Because you couldn't carry a lot of the meat because the meat would rot. And I don't want you to think that these were vegans at all. The caloric density is going to be in animals, and the animals would have been plentiful because they can break down cellulose and hemicellulose. Now, those are polyglucan carbohydrates as well, but they have a beta configuration for their bonding. And we don't have the ability, mammals such as us that are monogastric, to break down cellulose and hemicellulose. Animals that have that kind of gastric system that ungulates have, right? The multiple stomach ungulates and a functional cecum like in horses can break down cellulose and hemicellulose and use that to make glucose so that then what they can use their glucose for, of course, the synthesis of polypeptide and lipid, etc. Right? Humans can't take all of that carbon that's growing out in the field where animals like deer and elk and mammoths and aurochs and things like that can. So the early human evolution of the biochemical pathways was humans would prey upon the animals, would take down an animal, process the meat, consume the food. Maybe there was some cooking being done by then, 200, 300,000 years ago. Certainly there was the invention of fire or the discovery of how to make fire. And so the food would be consumed and then people would move on, right? So the kind of plants that we're growing that you might say, well, what about all the fruits and vegetables we consume? We consume a lot of fruits and vegetables because we have agriculture. So we make row crops of lettuce and radishes and spinach, all of those greens that we can grow in rows, and then we can harvest them and wash them up and put them in grocery stores. Or if you're like what I did for a long time, have big gardens, you wash them up and put them in the refrigerator, but they don't keep very long. And by the way, the same problem with the carbohydrates that's in all of those greens, lettuce and spinach, for example, celery, they're not going to be growing in rows. So you're not going to have a lot of that available during the evolution of the biochemical pathways in the mammalian gut, etc. They're going to be very sparse. And so you might be able to consume some greens in the diet, but you're not going to have the kind of greens that are going to be particularly palatable. Think about when you're roaming around in southwestern Colorado, the kind of green plants you're going to be encountering are not going to be the kind that you can digest very well. For one thing, they're going to have all kinds of thorns and probably a lot of substances that would be quite toxic, such as terpenes, that you wouldn't be able to consume even what might be some protein that could be digested from those plants. So it's going to have a high animal caloric content in the diet. And that is how our entire digestive system, utilization of lipids, carbohydrates, and proteins still exists because there hasn't been any heavy duty selection pressure because there hasn't been that long for the evolution, right? If that's how you look at the entire process of events, which is how I look at it. Okay. So that means the processing of the food that you're consuming in 2023 is dealing with biochemical and physiological biochemical processes that evolved when there was not row crop. Same thing with fruit. Now, we grow apple trees in orchards. We grow grapes in vineyards, right? We have apricots and we have oranges and we have all different kinds of melons and whatnot. Those carbohydrates, because there's a high level of sucrose in them, we can, of course, utilize. But you can't utilize it if those food sources are not readily available, again, in modern agriculture. So there may have been melons, some wild melons, some maniac growing out there, but it's not going to be in a one field where there are like kilograms of food for the entire clan to be able to consume along with the meat that they're killing. See, because those plants are not growing in monoculture. They're growing out there and competing with all the other plants that are growing wild. So one of the main things about agriculture isn't simply that we understood about nutrition and breeding, which itself is a fantastic human achievement. But we also started to learn about nutrition for plants. We didn't know anything about, nitrogen, phosphorus, potassium, and all the other macronutrients and micronutrients. But early farmers learned that some soil would grow plants better than others. And also the breeding made it easier to start breeding for plants that would they all would be harvestable, generating a harvest index that is a high edible biomass divided by the total biomass of the plant. You can breed for that when you're doing plant agriculture, but also so that you get plants that will come to fruition at the same time, right? So there's a temporal thing. So you want earliness so you can get as much crop in as possible for the next frost comes. You want plants that can take up nutrients very effectively and efficiently. So you learn about breeding for that and maybe occasionally poor soils as you're moving around. And also this whole aspect of earliness and then high biomass, right? High edible biomass. None of that was going to be available during the evolution of digestive system. So that's what we're looking at here. Right? So again, with long-term fasting, you're getting protein breakdown in the liver. You can generate glucose from the amino acids because of transamination reactions, running those keto carbon molecular species through the TCA cycle and then generating ATP directly, right? You also need that ATP for gluconeogenesis. So amino acid conversion in terms of the carbon can directly go into glucose because many amino acids are directly gluconeogenic. Some are ketogenic, as you know from your probably high school biology class. So you can synthesize glucose directly from protein, but humans cannot synthesize glucose directly from fat. So we don't have the enzymes of the glyoxylate cycle, but plants do and microorganisms do. So without the glyoxylate cycle, you cannot convert acylipid, that is fatty acid, which is the main form of triacylglycerol, into carbohydrate. So the liver has to deal with utilizing amino acids for that or organic acids like lactate, right? And the whole business of the lactic acid cycle that's generated then ultimately as an end product of glycolysis and then pyruvate to lactate in the bloodstream, lactate to the liver, gluconeogenesis from the lactate. Just keep that in mind. All the lipid you can consume does not get converted directly to glucose. We don't have the enzymatic machinery, we don't have the pathway for that. Now, you might wonder, why would that not evolve for humans to be able to take that? It's because mammals in general, and many animals, are oleaginous organisms. We're able to store, against a huge concentration gradient, a great deal of adipose, right? And that adipose is the best way to store carbon because it doesn't require water. So there's no water of hydration that you need for carbohydrate or protein. That means the mass of the body carrying that burden is going to be reduced because there's no water needed to store lipid, because lipid and water, because of the hydrophobic effect, water is going to hydrogen bond to itself, generating a force that will induce the lipid to stay as a lipid. You'll get an aggregation of the lipid in forms of oil droplets. And so because of that, the interaction between water and neutral lipid in particular, you're going to have a biophysical phenomenon. It doesn't require any kind of genetic regulation, just you're going to have a separation of the two layers, the aqueous layer from the lipid layer, right? And that's how adipose is generated, basically, just by keeping the adipose free from water. But I want you to understand that because lipids cannot, anytime you think about fatty acid utilization, it has to be esterified either to oxygen or something like sulfur. Thioester oxygen esters to be able to be mobilized, utilized, and then not converted to carbohydrate, but converted to those ketone bodies, acetoacetate beta-hydroxybutyrate, which can serve well. Now, those ketone bodies can then be used to make glucose, or they can just be directly pumped into the TCA cycle, generating NADH. NADH reoxidizes in the electron transport chain, running through the 5 complexes, last complex being what? The F9, F1 ATPase, making ATP in the mitochondria, sending the ATP out of the matrix of the mitochondria while bringing ADP and PI back in. Right? Okay, so that's a very quick summary of what's going on. But that's why if you ever want to understand why there's a difference in bioenergetics, as you start to go from the well-fed state, which most humans are these days because of the invention of agriculture, because of the fact that we know how to store food, process food, and we've made crops real high in, for example, sucrose. Those are what most of your fruits have, real high, sucrose. The human body doesn't really digest sucrose as effectively as just glucose. And so fructose is derived from sucrose. Doesn't go through glycolysis, it goes through fructolysis. And that is a main source of an increase in body mass because fructolysis is less efficient in carbohydrate metabolism than glycolysis is. We can talk about that later. So just so you understand that none of those foodstuffs you think about that would have been available, and the whole argument that you need all of this fruit and vegetable, really what you need is lower caloric density foods. You need to have longer periods of fasting to digest that food so that circulation can keep up with the trafficking of lipids as lipoproteins. Remember that you have lipoprotein trafficking to traffic lipids that are not soluble in aqueous. What's the serum? It's aqueous, right? So that whenever you see a lipid in circulation, it's going to be with something like serum albumin, or it's going to be with a lipoprotein fraction. Right? And all of that's a slow process to get it where it can be broken down, for example, via beta oxidation, even intracellularly, let alone when you think about total circulation. All of these are issues that have evolved very well in humans, very efficiently. So we store a lot of depot fat. And if we eat too much, and it's a chronic event every day for weeks and months and years, people become obese. And this was normally about the time that the rest of the enzymatic machinery that synthesizes nucleic acids and the storage of DNA and the telomere links becoming degraded, the ability to be able to replicate the genome, right, via DNA synthesis, and then to maintain all of the other enzymatic machinery as you age starts to decrease in efficiency. And so cells will quit dividing, and that Hayflick limit eventually is reached, and you start to go through senescence. And senescence isn't programmed cell death, and it's not necrosis. Basically, it's aging of the cell. And we age as our cells age. And after a while, those cells no longer function appropriately because they lose all the regulatory components. That is at the level of allosteric regulation of enzymes, but also at the level of genetic and epigenetic regulation of transcription, translation, post-translational modification, activation of those proteins, and turnover of those proteins. All of that starts to go to ground, and that's what ultimately leads to death. Now, the obesity, because of the high caloric density foods, never used to be a problem until people, maybe if they had a really efficient metabolic pathways, and they had a source of a lot of food throughout their lifetime, they wouldn't really become obese because they were also doing work, physical work during the evolution of these processes. But now people don't do too much physical work. They go to the gym to work off some of those calories, but they consume way more calories than they need. So they become obese earlier. So all the diseases that we see that are the primary causes of high morbidity and mortality, I'll just name two of them, cardiovascular disease and cancer. Those are diseases that are directly correlated, not caused by, but correlated, or I'll just say linked to obesogenic environments. And that's a major focus that I tried to do in authentic biochemistry. So I'm saying this, and I feel like I'm preaching to the choir because I know many of you, if you've heard my lectures before, know this story I'm telling you. It's not a story, it's not a narrative. You look at the evidence of what the system does in terms of regulation, and then you look at the pathophysiology, and you can then construct why and how the metabolic pathways evolved to deal with a level of limiting calories per day, not excess calories per day. And so not so much the consumption of what given foodstuffs you have. You need some roughage. Roughage simply means you don't digest it, so you extract no energy from it, right? So you can't live off of roughage. Beta 1,4-glucans, beta 1,6-glucans. You can't digest any of that, but roughage is necessary for digestion, of course. But in terms of bioenergetics and in terms of generating new cells and in terms of maintaining living systems, you need readily digestible carbon sources. And those are going to be protein and lipid and sure, some carbohydrate, but it's going to have to be in the right configuration. And that's the whole point of the last 20 minutes that I've been talking about this. Right? And that's what becomes corrupted. So you need some vitamins from the diet, and vitamins being essentials that are not synthesized, usually acting as some kind of prohormone or to regulate some metabolic reactions, something like biotin be involved actually in enzymatic reactions. But many of the other essentials, for example, pantothenic acid, well, that's a precursor to coenzyme A. Right? So we have essentials in the diet, which we do get solely from plants. Think even about the two essential fatty acids, linoleic and alpha linolenic acid, two 18-carbon fatty acids. One's 18:2, the other's 18:3. 18:2 fatty acid. The last double bond is 6 carbons away from the methyl terminus, so it's omega-6. That's linoleic acid. And then the 18:3, its terminal double bond is only 3 carbons away from the terminus, so it's an omega-3 fatty acid. Those are essential in the diet, and we can get those from plants because we can't. Human cells don't synthesize those essentials, just like they don't synthesize a lot of the other essentials we may get from plants, or we could indirectly also get them from animals. Some of these, of course, because animals require them too, because animals require those essentials in their diet. Because they're animals, right? So they're not able to synthesize. And no, the bacteria in your gut are not able to keep up with all of the requirements for all the quantity of essentials that you synthesize and utilize in your body. And again, you don't synthesize the essentials. You synthesize from the essentials those components, such as taking linoleic or alpha-linoleic acid, making arachidonate for linoleic acid, and icosapentaenoate and docosahexaenoate from alpha-linolenic acid. So those are the fatty acids, for example, just using those as an example, why not, are what are most important for metabolism in the human, okay? And in mammals in general. All right. So I know I'm talking really fast here, and I'm going through this pretty fast, but hopefully, first time I've done a video lecture in a while, so hopefully this isn't a total bust. But if we were in lecture hall, maybe there would be some questions now, maybe not, because a lot of the things I'm mentioning, maybe you've never heard before and you haven't thought it through. But that's the difference between having authentic biochemistry as a knowledge base, and rather than having a textbook, let's do chapter 1, chapter 2, chapter 3, where there's a lot of analysis, but very seldom is there solid, sequenced synthesis. And that's what I do in authentic biochemistry, because I've been doing this a very long time, right? And the synthesis comes with an understanding of the system, right? The living system, which is fundamentally focused on biochemistry to physiology. Now, the fundamentals are not genetics. genetics play a role in the transmission of all the information from cell to cell? Absolutely. But biochemistry is involved in all the genetic aspects of living, right? All recombination, DNA synthesis, DNA repair, all of that are biochemical events. Right? That's the foundation of living systems. And where does biochemistry come from? An understanding of physics, an understanding of chemistry, and an understanding of biology. So you need all of that to be able to grasp biochemistry, right? I think I'm going to stop there. I think I probably spent a long enough time for this video lecture, and it's been a while because I was going to talk about glycogen and the utilization of carbon here, as you can see from this slide. But I think that would, I would rather leave that until next time. So, Dr. Dan Guerra in a schoolhouse in southern Colorado, saying, as soon as I can remember how to shut this whole thing down. Yeah, right there. End show. Okay, why didn't it end? Well, odd stuff here. There it is. Okay. Yeah, so let's go back to where we were when we started this whole process, which is this here. Yeah, that's right. And now I'm just looking for the goodbye. We'll get rid of the share button. And there's a record button. Oh, wow, look at that. I didn't even know we had that. Okay, I will end here. This is Dr. Dan Guerra. Tried to dress up for you a little bit with my fancy shirt. Hopefully you will enjoy this video lecture. It was very impromptu. I just thought, well, I'm going to go ahead and do it. I'm going to get a better camera soon. So I'm using older camera equipment and older audio equipment. But all of that's going to come up when I set up my new studio, which will be in about a week or so. So have a very revered Memorial Day weekend for all of those who have died to protect our country in the United States. For those of you that aren't in the United States, I think we're all probably involved in that one way or another, and all the wars that had happened in the past. That's what we are honoring on Memorial Day weekend in the United States. And so with that fervent understanding, this is Dr. Dan Guerra from southwestern Colorado saying bye for now. I thought. All right. This is different than last time, so let's get back to this. There it is finally. All right.